Now that we've spent some time talking about basic rules of inheritance, we're going to spend a little bit going over how gene expression itself is controlled. This is generally split into three major topics: the control of gene expression, biotechnology or the cloning of plants and animals, and the genetic basis of cancer. In the interest of time, we're not going to spend much on biotech itself. If you're interested in biotechnology, there are courses specifically in this topic that you might want to take. We're going to start with this idea of how is expression controlled. When we talk about genetic information and how it flows from genes to proteins, that is basically what we're discussing, the expression of a gene. Gene regulation is the turning on and off of genes, and this can help organisms respond to environmental changes. You might have a situation where having a gene turned on is beneficial to you in one environment, but is bad for you in another environment. So you want to be able to control when the gene is on and when the gene is off. And we'll talk about some mechanisms that organisms use to try and do this. We're going to start with simple mechanisms that are primarily present in prokaryotes. Here, genes for unrelated enzymes are often controlled together in a unit that's called an operon. There's generally a regulatory protein that is going to help bind to control the sequences of DNA all together. The basic structure of a gene contains three parts: a promoter, which is going to recruit RNA machinery to make an mRNA, the coding region, which contains information about what the protein this mRNA encodes for will be, and the termination sequence, which says that the mRNA is going to be complete and the entire gene has been copied. Operons have a slightly different structure. They're usually composed of multiple genes that are put together. The first gene is a regulatory gene. This gene has its own promoter, has its own coding sequence, and its own termination sequence. And this is going to encode the regulatory protein, which will control the rest of the operon. The operon is then made up of a promoter, multiple coding regions for each gene that is going to be controlled, a termination sequence, and a special sequence called the operator. The way that an operon functions is that when the regulatory protein is produced, it can interact with the operator. Under some circumstances, it will bind the operator directly. This prevents RNA polymerase from being able to bind the neighboring promoter, which means that none of the genes downstream of the operator can be expressed, and no mRNA will be made. In order for an mRNA to be made, something has to change that is going to cause the regulatory protein to be released. Usually this is environmental change. When the regulatory protein is released, this allows RNA polymerase to interact with the promoter, and this means that all of the downstream genes can now be expressed. The lac operon is a famous example of this. We talked previously in the course about how lactose is something that most people have a difficulty digesting, and that is because it requires the expression of specific enzymes to break down. The way that the lac operon functions is that the regulatory protein is active. This means that under normal circumstances, when lactose is not present, it will bind the operator and prevent the expression of any lactose utilization genes. However, if lactose suddenly becomes present in the environment, this will inactivate the repressor, allowing RNA polymerase to interact with its promoter and thus express all of the lactose utilization genes. This means that these genes are only expressed when lactose is around, so that the prokaryote does not waste time or resources expressing these genes when it doesn't need to. This type of operon is what we like to call an inducible operon. This means that the default state is off, but that expression can be induced by the presence of something in the environment. The other major class of operon is a repressible operon. In this case, the default state of the operon is on. The presence of a specific molecule in the environment can turn off or repress the operon. The trip operon is a classic example of that, where presence of tryptophan in the environment turns off the downstream genes. Now that I've presented some information about how genes can be controlled, at least in simple organisms, I want to impress on you how important this is in organisms like you. A zygote, for example, is a multicellular organism that is made up of individual cells. These cells usually have specific identities. They might be neurons or muscle cells or cells in your skin. This happens through a special process called differentiation, and that allows cells to specialize their structure and their function. We're going to watch a short video where we basically watch this happen in a fly embryo. The embryo starts as a fertilized egg and is going to develop and eventually differentiate to the point where it is a hatchable larva. If you look really closely, you can even see it walk off the screen at the end. So, how do these cells know what they're supposed to become and where they're supposed to be? The answer to that question is expression. Each cell type has to maintain a specific set of genes that are on and specific sets of genes that are not, and that is what makes a cell an individual. So all of your cells contain every possible gene in your genome, but they don't use all of them the same way or at the same time. Let me give you another kind of example. Here we're going to look at the nervous system of a fly. In the picture to the right, you can see the nervous system is lit up with this green color. This is called GFP. GFP is a green fluorescent protein. Basically, the way this kind of indicator works is that we have its standard gene structure. And I told you that promoters tell us some information about where genes should be expressed. So what we can do is we can take the promoter of a gene and we can attach the coding sequence for GFP to it. Now, GFP will be expressed anywhere that original promoter says that it should be. This allows us to look where specific genes should be expressed. Oftentimes when we think about something like neurons, we think that all neurons are going to be similar to each other, but that's not the case. For example here, one promoter on the left only shows GFP expression in the optic nerves. These are nerves that control the fly's ability to see. Another promoter on the right only shows expression in the olfactory nerves. These are nerves that are important for allowing fly to smell. Different types of neurons make different neurotransmitters and express different genes, and that's what allows them to do the job that they need to do for the brain. There are multiple different types of GFP which allow us to look at multiple promoters at the same time, or to get creative and paint pictures if you really want to. When we apply different colors to different promoters across the brain, we can see that there are a lot of different subsets of neurons that have very specific expression. And this likely means that they also have different functions. Okay, sure, Dr. Sitnick, that's cool, but what about mammals? I'm not a fly. Well, mammals aren't any different. This is a picture of the hippocampus of a mouse, part of the brain, that has also been lit up using this kind of technology, and you can see that there are a lot of individual neurons that have very different colors, and therefore very different gene expression patterns. So, why did I show you all of that? Do I really want you to know exactly what a fly brain looks like? Well, no. What I want you to get out of this is that the differences between cell types are not due to different genes being present. All cells have your entire genome. The difference is actually due to selective gene expression, and that's what actually creates individual cell types within your body. Not everything about gene expression is controlled just by promoters. For example, there are special genes called homeotic genes that are master control genes. They're important for determining your body plan, and they control tons of other genes downstream. So let's talk a little bit about them. For example here, what you can see is that there are multiple of these homeotic genes: IAB, PB, DFD, SCR. Don't worry about needing to know all of the names. Each one of these corresponds to a specific part of the developing embryo in the video we saw before. Some of them are going to control the production of the head, some the thorax, some the abdomen, and some the tail. They will start telling cells in these areas, here is what you are supposed to be. The thing about homeotic genes is that they're actually very conserved. So here are homeotic genes that can be found in the mouse. Colors that match represent the same gene. So for example, IAB, the head regulator, is also found in all of the Hox genes for mice. And you aren't particularly different. Here is the way that those Hox genes map onto your own body. Okay, so they exist, but what do I mean by they are master regulators? Well, let's say we take a Hox gene that's supposed to be in the thorax. One that, I don't know, maybe controls whether or not you produce legs. And we decide that we want to express that in the head. Well, here you go. This is a fly that is expressing the thorax Hox gene inside the head area. It's called antennapedia, and you can see that instead of antennae, it's developed legs. We can also do things like say, okay, we want to repeat one of the thorax segments, the one that contains the wings. So we're going to tell the subsequent segment, the one that comes after it, that it should look the same. Well, here you go. That's called bithorax. Something as simple as misexpressing a single Hox gene is enough to create major body plan differences. Now, again, you might say, okay, sure, that's a fly, but I'm not a fly, so what about people? Well, you're not any different. As you might imagine, major body plan differences in human beings can be very scary to look at, so we're going to limit our discussion to things that control the way your arms develop. There are 5 Hox genes that are important for developing the forelimb in mammals. Notice how they are in order. HOX9, which controls the shoulder, is going to come before HOX10, which will control the upper arm, which will come before HOX11, which will control the forearm, comes before 12, the wrist, comes before 13, the fingers. These Hox genes need to be in order and expressed properly in order for your hands and arms to develop properly. So, what would happen if we had a mutation in HOX13? What might be affected? If you said the hands, you would be right. This is a mutation in HOX13. What you can see is that the arm up to the wrist looks pretty normal, but the fingers are not able to differentiate properly, and a lot of them are fused or missing altogether. So mutations in HOX13 prevent proper formation of the fingers. What about a mutation in HOX11? What might happen then? If you said the forearm would be affected, you would be correct. So if we look at a schematic of the arm, you can see that the ulna and the radius are generally separated and located in the forearm after the elbow. What happens in HOX11 mutations is that the ulna and radius usually don't form properly. The most common form of mutation is where they are fused together. This creates an issue where individuals have limited mobility and they aren't able to flip their wrists outwards. Here, hold your hands in front of you and see how the ulna and radius have to pass around each other in order for you to turn your hands upward. Imagine what it would be like if the ulna and radius were fused and you couldn't do that. So how do Hox genes do all of this? Well, the answer is that they are genes that encode something called a transcription factor. Transcription factor is a protein that promotes the binding of RNA polymerase to a gene. They do this by binding near the promoter of target genes to help turn them on, and this is why they can control multiple genes at the same time. You don't need to know that there's HOX10, 11, and 9 and all of that, but you do need to understand that controlling the expression of multiple genes at a time is what a transcription factor does, and that's what a Hox gene or homeodomain gene is. Alright, so we've got operons in prokaryotes, and so far we've got Hox genes in eukaryotes. So what other ways can we control gene expression? The easiest way is by controlling the structure of the DNA itself. So in order for RNA polymerase to get into the DNA to make an mRNA, it has to be able to get access to that. We've talked about how chromosomes can be condensed or decondensed. So controlling whether or not a chromosome is condensed can help us control whether or not a gene is expressed. Using the structure of a chromosome to control expression is called DNA packing. In order to be expressed, DNA has to be unwound, and transcription factors can help do this. But if the transcription factor can't get in because it's too tightly packed, well, no expression is going to happen. We're going to talk about 2 specific examples of DNA packing, and that's going to be X inactivation and epigenetics. Sometimes your book will connect epigenetics to inheritance, but we'll talk a little bit about why that is difficult to do. So let's start with X inactivation. How many X chromosomes do you need to be a fully functioning human being? If the answer you said was 1, you'd be right. Males have 1 X chromosome and they're perfectly fine. So 1 X chromosome must be enough. Well, it turns out that 1 X chromosome is actually the right number, for most things anyway. So having 2 complete X's active is a bad thing. In order to solve this problem, there's a process called X inactivation. This process is pretty random, and basically what it says is 1 X chromosome is going to be hypercondensed and essentially silenced in each cell. You can see the condensed X chromosome in any cell that has multiple X's. It's called a bar body, and it looks like a really dark dot. This is going to happen anytime extra X chromosomes exist. So for example, in a normal female, there should be 1 active X and 1 bar body in every cell. In a normal male, there shouldn't be any bar bodies, because there's no extra X's. In a Klinefelter male who's XXY, you'd see an active X, a Y, and a bar body, and in a triple X female, you'll see 2 bar bodies. So see, every extra X chromosome is going to be silenced. This is a process called dosage compensation, and it guarantees that only 1 X is ever going to be expressed. Now, we talked about how there are a lot of genes that are located on X chromosomes. So what happens then? Let's talk about an example. There's a gene located on the X chromosome in cats that determines whether or not a cat will be black or orange. The orange version of this gene is dominant to the black version. Now, if you're a male cat, whatever copy of the X chromosome you have is the color you'll be. So if you have the dominant X, you'll be orange, and if you have the recessive X, you'll be black. It also stands to reason that if you're homozygous for either of these X's and female, you'll be that color as well. But the real question here is, what happens when you're heterozygous? Under normal circumstances, we would expect this individual to be orange, right? Orange dominant to black. But that's not what they look like at all. Instead, they look like this. This is a tortoiseshell or calico cat, which generally has patches of orange or black all over its body, and 99.999% of them are going to be female. The reason for this is that this color pattern is caused by X inactivation. So this individual is heterozygous for these two traits. Cells that inactivated the black X will look like orange patches, and cells that have inactivated the orange X will look like black patches. So let me ask you a question. I said that most of the time these individuals are going to be female. Can you think of a scenario where you could have a male cat that would look like this? What about a Klinefelter male? They've got two X's, right? So they should be able to do this as well. This means that calico males are almost always sterile, so it's not a good idea to buy one from a breeder. Alright, so what about this idea of epigenetics? Epigenetics is the study of changes in organisms caused by modification of gene expression, rather than the genetic code itself. It's a hot term right now. The idea here is that everything that you're exposed to during your lifetime is going to have an impact on the way your genes are expressed. Things like smoking, or taking certain drugs, or how much money you have, or what kind of food you eat. There are lots of different ways for epigenetic events to occur, but the most common one is methylation. Methylation is the addition of a methyl group onto a C nucleotide. When methylation is present, this causes the DNA to tightly pack and silences the gene that is involved. When they're acetylated, or the methylation marks are removed, this opens up the DNA and allows transcription to happen. This is an easy way for you to silence a single gene if you need to, and usually happens in response to environmental effects. Some methylation marks cannot be removed and become permanent for the individual they occur in. These are often things caused by long-term environmental exposure, like famine or smoking. Up till this point, everything we've talked about is how to regulate a gene before mRNA is produced. However, there are quite a few ways we can regulate genes after mRNA is produced. One of these has to do with this major conundrum. We only have 21,000 genes, but we make more than 100,000 different proteins. How is that even possible? This relates to an idea we've already talked about, which is splicing. The coding regions of genes include two different sequences, exons and introns. The exons are going to include information that is needed to make a protein. The introns are going to be removed and the exons put together to create the final protein. But here's the deal. We can actually change which exons we want to have included. For example, we might decide to skip exon 2 entirely. This allows us to use one gene to make two different proteins, one that includes exon 1, 2, and 3, and one that only includes exons 1 and 3. This process is called alternative splicing, and it's actually the rule and not the exception. More than 90% of your genes are going to go through alternate splicing events, and this allows a gene to produce multiple proteins. Which splicing events are going to occur will depend on which protein is needed at the time. Other ways that we can regulate transcripts relies on another strange attribute of DNA. Only about 1.5% of your genome actually encodes for proteins. Most of your genome is actually made up of things that, for the longest time, people thought did nothing. Back in the 70s, this material was thought of as junk DNA, or kind of just leftover stuff from previous organisms. It wasn't until the human genome was published in 2001 that we realized that the vast majority of our DNA was this so-called junk. It wasn't until a few years later that we actually started looking at what junk DNA might be doing, and it was determined that most of this DNA actually does have a function. The vast majority of this DNA is transcribed into non-protein coding RNAs, and these are frequently used to control the expression of protein coding RNAs. An example of that is something called a small single-stranded RNA, or a microRNA. These RNAs can be complementary to mRNAs, and this allows them to help regulate them. So let's talk about how that works. Let's say that we have a microRNA complex. This consists of two proteins and the microRNA itself. This complex is going to look at mRNAs that are produced within the cell and look for complements. If there's a perfect complement, this is going to target the mRNA to be destroyed. This is because double-stranded RNA is not something that generally occurs in eukaryotic cells. This is usually something that is seen in viral genomes, and it's something the cell wants to protect itself from. If there's a partial match, instead, this will prevent the mRNA from being translated into a protein. So we can use the expression of microRNAs to control whether or not an mRNA ever becomes a protein. So today we've talked about a lot of different ways that genes can be regulated. Before transcription, they can be controlled by operons in prokaryotes, which will determine whether or not sets of genes can be expressed based on environmental factors. Promoters determine basically where a gene should be expressed. Transcription factors like Hox genes can recruit to the promoter to increase expression. And DNA packing, whether or not the DNA is condensed or open, can determine whether or not a gene is expressed. We talked about two examples of this, exon activation and methylation. After transcription occurs, we discussed two different ways the genes can be regulated, either by alternative splicing, which determines which exons will be kept in the final mRNA, or by microRNAs, which can determine whether the mRNA should be degraded or prevented from being translated. All of these things are going to work together to help determine which genes are going to be expressed and therefore what a cell's identity is going to be. Now let's use some of this information to talk about something that people are actually interested in, and that's the genetic basis of cancer. Essentially what cancer is loss of control. We discussed previously that there are a large number of genes and events that are going to help control the cell cycle. When mutations occur that remove these controls, that's when cancer happens. In general, these mutations are going to happen in one of two places. In a gene called a proto-oncogene. These are genes whose proteins tell cells to divide, or in genes called tumor suppressor genes. These are genes whose proteins tell cells not to divide. Let's think about an example. Here we're starting with a proto-oncogene. This creates a protein that stimulates cell division. Under normal circumstances, this cell will only divide when it's supposed to. If we have a mutation that's within the gene, we might have a situation that when the proto-oncogene is turned on, the protein is super effective. This means normal amount of protein, but increased cell growth. We could also have a situation where we create duplications of the proto-oncogene. Under this circumstance, when the gene is turned on, we will produce the normal amount of growth-stimulating protein for each copy, creating an excess. And we could also have a mutation in the promoter of the oncogene, causing it to be active when it is not supposed to be. This creates the normal stimulating protein, but it might produce it when it's not supposed to be made, or it might produce too much of it when the gene is active. All of these situations would lead to overgrowth in the cell and cancer. Now let's think about what happens for tumor suppressor genes. These are genes that produce proteins that inhibit cell growth. Under these circumstances, when the tumor suppressor gene is on, it's going to prevent cells from dividing. However, if we have a mutation in the tumor suppressor gene that creates a protein that is no longer functional, this prevents it from repressing cell division and allows cells to divide uncontrolled. Normally when we talk about cancer, we have a combination of these kinds of mutations. So when we talk about stages of cancer, we're actually talking about the number of mutations that are leading to changes in the cell cycle. So one mutation is not enough to cause cancer. 2 or 3 mutations would be stage 2 or 3 tumors, and stage 4 would be when the cell finally becomes malignant. Understanding that changes in the regulation of expression of genes or their functions is what underlies cancer is important for treating it. Cancer is the second leading cause of death after heart disease in most industrialized nations, and this is because it can arise from mutations caused by all kinds of environmental factors. Anything that changes DNA can make cells cancerous, and these are called carcinogens. Anything to reduce exposure to carcinogens and making other lifestyle choices can help reduce cancer. Cancer sometimes results from mutations in genes that control cell division that can be inherited. An example of that is BRCA1. This is a tumor suppressor gene, and its job is to repair DNA damage and prevent tumors. Mutations in BRCA1 are associated with prostate cancer, breast cancer, and ovarian cancer. That doesn't mean that BRCA1 mutations cause cancer, but individual carriers of these mutations are much more likely to develop it over the course of their lifetime. For example, breast cancer, by the age of 50, only 2% of the general population will have developed, but individuals with BRCA mutations, roughly 33 to 50% will have developed by age 50, and 56 to 87% by age 70. This is because once you have one cell cycle mutation, it's much easier for you to get others.